Alright, so we've got a couple sources of variation, which clearly I'm not particularly happy about in this picture. One of them was... Guys, it's not that funny. Okay. One of these sources of variation is beam damage, as Aaron mentioned. So this was actually left in a Zane scan for several hours, which could have caused a redox reaction. Our selenium selenate could have been reduced, sodium selenate could have been reduced to elemental selenium, and that could explain that. Also, there were smaller levels of selenium in our mustard seeds, so it's possible that with a different beamline, we would have been better able to analyze the speciations of selenium. As well, we just actually got a phone call from Bulk Barn right now. Our Brazil nuts came from Bolivia, and depending on where they're from, the selenium composition of the soil can greatly change the amount of selenium in your product. So our Brazil nuts actually were found to have very low selenium. Now, we're talking about Brazil nuts. Where was the Zanes for the Brazil nuts? While we were running that, we had a beam dump, so we lost all our Brazil nut data. It kind of crushed our nuts, so it kind of hurts, but again, not happy about the sources of variation. So there you are.
Okay, so through the experiment that we conducted, we were able to conclude that the scans of each tablet that had labels that mentioned that they contain selenium yeast had slightly different speciation of selenium present in each of them. And also we found out that the Zanes for one-a-day supplement showed us that the Zane graph of the one-a-day supplement was actually more similar to the elemental selenium than what it mentioned, which was the sodium selenate.
So the future developments of this type of research can include the bioavailability of both the inorganic and the organic forms of selenium. And this is because while we do know that inorganic selenium cannot be as bioavailable as the organic forms, we are wondering whether this leads to toxicity or how the body uptakes it differently than the organic forms. And then also, we do know that there is a really, really fine point in the spectrum of too much and too little selenium that's healthy. So if you don't have enough, you can be at a high risk for perhaps developing cancer. And at too much, you can be at the same amount of risk. So we are wondering why that possibly is.
And mentioning cancer, there have been many studies done on the benefits or maybe detriments to selenium in a diet if you're concerned about cancer, which we all are. So yeah, there's lots of differences in people's results in experiments that involve cancer. So we are wondering if there could be more research done to determine maybe different speciations of selenium and how they relate to cancer. Maybe they're carcinogens, or maybe they help in preventing cancer. Also, for the supplements, we notice a discrepancy between the measured weight of the supplement and the listed mass of selenium which is present inside the supplement. So that leads us to believe that there are significant fillers inside of these supplements, which we'd like to investigate further, maybe how they react with different speciations of selenium, or just how their general health effect on humans.
All right, so we just like to take a minute to talk about our students on the Beamline experience, and we're just going to give you one thing that we learned. So Aaron, would you start us off?
I very much learned the importance of preparing samples in the correct manner.
Rajan?
I learned that attitude is everything. So while we did hit a roadblock in our peanut study, the way that we took it in stride really helped us to do a whole nother project on top of that.
I think I learned that caffeine is really important to a researcher. Not to contaminate your samples by touching your morning cup with your gloves.
Yeah, I also learned the caffeine thing, but I think what I really found out is there are way more questions than answers out there. And also, Aaron can strike a mad pose. Sarah?
I learned a lot of important science things, but I also learned that when you stay up till 3 a.m. watching a meteor shower and then 3 a.m. the next night doing homework, your brain isn't really at a prime place for doing research the next day.
I learned the importance of asking questions. With so many great minds here, you just have to ask, and they're so willing to share with you, and that's really how you enhance your own knowledge.
I think we really took to heart words of wisdom from Miss O'Connor, work hard and play hard. We had a lot of information to get through, but we definitely took some really good breaks, and that actually helped us power through a lot.
I came to really appreciate the universality of science itself, and now we are unfortunately or fortunately part of a group of scientists that can also make a bunch of nerdy jokes that only we seem to understand. So, life's good that way. And yeah.
I learned an analogy that Tracy told us a few days ago about saying that we want to cure cancer, so as a result we're just going to move this little drop of solution into a petri dish. And it taught me that every step is important in the overall process, whether you can see it directly or not.
I learned from Tracy her #1 rule is not to lick the samples, even if they appear to be peanuts and edible, they're still samples, so don't eat them.
Through this experience, I learned